billion years ago, our ancestors were swine. How did life on Earth become so diverse, using many of the same basic genes?
If you buy 1 million lottery tickets, the chance of hitting the jackpot is pretty good.
The directions for a human being are written in code, 3 billion letters long. These instructions tell our bodies how to live, how to grow, how to die. Researchers call this code the sequence.
Here's a riddle. What's as similar as 2 peas in a pod, but as different as night and day? The answer? Most any 2 forms of life. At the molecular level, the difference between you and your neighbor, or you and me, is a matter of a few thousand genetic letters. And we have found that the difference between humans and our first cousin, the chimpanzee, is a mere 1.3%. Everything from a bacterium to a scientist is made from different arrangements of the same genetic material. How life forms evolved and diversified continues to mystify and astound us. It is a business of science, of course, to dispel mysteries, as it continues to do, but the wonder lingers on.
We understand that animals evolved from a single-celled ancestor, probably beginning somewhere between 700 million to 1 billion years ago. And the buildup of complexity of larger organisms, greater numbers of cell types, more complex behaviors, more elaborate structures, organs, etc., all this we're trying to trace by studying the genes that govern the formation of those structures in living animals today.
To get started, Dr. Carroll and his team turned to the scientist's workhorse, Drosophila, the fruit fly. Much of the fruit fly's DNA is the same as ours and that of other animals.
No matter how different animals are in size, shape, composition, whatever we might see as dramatically different visually, fundamentally, the genetic logic and the genes that govern body formation are quite similar.
But if all creatures have similar genes, how can we be so different? The answer? Mutations, the agents of evolution. These are changes in the structure of a gene that might be caused by a virus or some form of radiation, among other things. Such changes that can have major effects down the line, providing they make it past the natural selection process.
About natural selection. In his book, Origin of the Species, Charles Darwin introduced the theory of evolution by natural selection, a process through which species exhibit gradual change caused by mutations over thousands of generations. If these changes create a survival disadvantage, they will eventually be selected out. If they provide a survival advantage for the organism, they will be passed on. For example, the survival odds for a butterfly in a green and brown landscape will be greatly increased by a mutation that gives it green and brown wings. Such changes within each group of a species build up over time and are inherited by future generations. This is known as biological evolution.
I think the butterflies are a microcosm of all nature, that because they have radiated into so many different types of forms, because they have so many different interactions with their environment.
The butterfly demonstrates a common theme, the same genes expressing different purposes. Carroll and his researchers focused on one specific gene that they knew had an ancient evolutionary history.
We understand that a gene called distillus, which is dubbed that because it's involved in the development of the distal, or sort of the part of the structures in the animal that stick out, that distillus gene is almost as old as animals themselves.
That gene has been retained for hundreds of millions of years in all types of organisms, including humans, and it has expressed itself in many different ways.
We use it to build all sorts of structures, for example, in our facial structure as well as in our limbs and all that. So some of these genes have a very, very long history, but within any particular line of evolution, they have some specific things that they've been used for. So the critical mechanism that goes on to create novelty in animals is the using of genes in new places in the body.
Some differences occur in the way the genes express themselves.
Genes carry information to each cell in a living organism, directing it what to do, how to behave, and what kind of structure to grow into. It is the genes within each cell that directs it to develop into a blood cell, or a brain cell in humans, a wing cell or an antenna cell in butterflies. They tell the cell what proteins to make, and some control gene expression profiles.
Understanding genetics is central to understanding diversity because at the absolute basic level, what makes a cat and a dog different or a butterfly and a fruit fly different is encoded in their DNA.
And the same is true for all creatures, including the lowly things that crawl upon the ocean floor. Researcher Nipam Patel at the University of Chicago has been studying how crustaceans, like lobsters, shrimp, and even sand fleas, are continually evolving. Like Sean Carroll's butterflies, these creatures, which all belong to the arthropod family, have developed pretty amazing diversity among their own kind.
Our main focus is understanding how they generate segments, which we know that they all do, and that comes from a common ancestor that they had, and then how they specialize their appendages, because it's these appendages or arms that they have that makes them so successful. And so we're interested in the genes that allow them to specialize these different appendages to do different things.
Arthropods have very unique body types. First, they wear their skeleton on the outside, an exoskeleton. This makes it easy to see how their body is divided into repeating segments. So if humans share most of the same genes as all living creatures, including these tasty fellows, how do the genes that create segments in arthropods correlate in humans?
So if you think of something like a millipede or a centipede that you see walking on the ground, you can see that it's made of repeating patterns. And those are the segments, the body segments. And usually most arthropods have a pair of appendages on each of those segments. We have an internal skeleton, so it's a little harder to see the repeating patterns that we have. But they're visible, for example, in the pattern of ribs that we have. It's a theme that biology uses over and over again.
Again, old genes learn new tricks. This reinvention helps each animal develop the specialized appendages it needs to adapt to its own unique environment.
It's believed that in the sort of most primitive type of crustacean that existed hundreds of millions of years ago, that most all of the appendages of the thorax were all the same, and they were used for swimming. But then as evolution proceeded and the crustaceans diverged and evolved, some of the crustaceans specialized their appendages sort of up near the head to actually use them for feeding as well.
Dr. Patel showed us some intriguing examples that demonstrate how these insects of the sea have developed in so many diverse ways.
So here we have a crustacean that's familiar to everyone, a lobster. And this is an example where several of these thoracic segments have been modified for use in feeding. So with something like a lobster, you have the ability to walk away, snap at something, tear up the food, and eat it all at once.
Patel and his co-workers have identified one of the areas on the genome responsible for the segmentation.
So one of the best examples is a set of genes called the homeotic genes, or the Hox genes. And this is a complex of genes that's found actually in all animals. And what it does is control what we call regionalization of the animal. It makes different parts of the body different from one another.
Okay, so I have a brine shrimp that's stained with UVX.
That looks pretty good.
So you can see it perfectly correlates with where the change in the limb morphology is. And
UBX, or ultrabithorax, seems to govern the specifics of segmentation in lobsters and brine shrimp. In humans, the UBX gene appears to control how our spine develops, just like a lobster's body develops in segments.
About human segmentation. The human spine is made up of 33 bony segments, specialized into 5 different types of vertebrae. Cervical vertebrae are flexible, but strong enough to hold up the head. Thoracic vertebrae are designed to hold more weight and attach to the ribs. Lumbar vertebrae are responsible for holding up the majority of body weight. Sacral vertebrae are compressed and fused together for support. And coccygeal vertebrae form the tail-like bone at the base of the spine.
So just like in our cousins, the crustaceans that use the UBX genes to specialize appendages, humans use the UBX family of genes to specialize our vertebrae.
And this gives us a really, I think, an increasingly clear understanding of how it is that evolution has taken probably a relatively simple organism that existed hundreds of millions of years ago and then generated an enormous diversity of life as we see it on Earth. And it's remarkable to think that that's happened from, a common set of rules and of genes that existed.
The Secrets of the Sequence teaching materials were developed at Virginia Commonwealth University, with funding from the National Academy of Sciences and the Pfizer Foundation. The original public television series, Secrets of the Sequence, was produced by Ward Television, with funding from Pfizer, the Pfizer Foundation, Oracle, and the Council for Biotechnology Information. Special thanks to member institutions of the series advisory board, consisting of Virginia Commonwealth University, Harvard University, University of Wisconsin, University of Michigan, University of California at San Francisco, and the MRC Laboratory of Molecular Biology, Cambridge, England.